Hello and welcome to our 2 days webinar about how to automate your liver perfusion and to reliably and reproducibly isolate primary hepatocytes. My name is Philipp Schlärmann. I'm working as a global product manager at Miltenyi Biotec and I'm responsible for the sample preparation portfolio. A short disclaimer before we start the presentation. Let's come to today's agenda. I will start off this webinar by showing you how important sample preparation is for the outcome of your experiments. Then I will explain you the GentleMax perfusion technology in detail and show you some comparison data between conventionally isolated primary hepatocytes and hepatocytes isolated with the GentleMax perfusion technology. Afterwards, I will sum up the findings and give you an outlook. Let's start smart and have a look into the importance of sample preparation. So why is sample preparation so important? Or in other terms, why does sample preparation determine the fate of your experiments? If you have a high quality input to your experiments, you have a high potential also to generate accurate and reliable results. If you have low quality input, you can be sure that also your results are inaccurate and unreliable. And that's why it's important to have a very good sample preparation to lay the basis for good and reliable results. If it comes to our products, you can be sure that we always aim for standardization and reproducibility. All of them are easy to use. We want to have as much automation as possible, and especially if we talk about our GentleMax technology, the automated dissociation of tissue, they are always gentle to cells. So how does the GentleMax technology of automated tissue dissociation and homogenization work? We are combining two different aspects, mechanical disruption and enzymatic digestion in one device, the GentleMax octodissociator with heaters. The mechanical disruption is performed by our unique GentleMax tubes. The enzymatic side is done by our easy-to-use tissue dissociation kits, supplying all the enzymes, buffers, and reagents you need for your dissociation. At the moment, we offer more than 20 different out-of-the-shelf, ready-to-use tissue dissociation kits for a vast majority of different tissues and different species. So let's have a more precise look on the mechanical disruption side. Our GentleMax tubes. These tubes ensure sterile sampling handling and are single-use. We offer two different types of tubes. Here on the left, the GentleMax C tubes in purple, and on the right, the orange GentleMax M tubes. The purple GentleMax C tubes are for cellular applications. So basically, whenever you want to obtain viable single-cell suspensions from solid tissue samples, you will use the GentleMax C tubes. On the other hand, we have the orange M tubes made for molecular or microbiology applications, and these tubes really homogenize solid tissue, so you can end up with RNA, proteins, DNA, intracellular bacteria, or viruses. Let's have a more precise look on the dissociation of liver samples with the GentleMax technology so far, which means before the release of the GentleMax perfusion technology. We offer our tumor dissociation kits, one for mouse, one for human. These kits work with pre-installed programs on the GentleMax octodissociator with heaters and are using the purple C tubes. By using this, you can get nice single-cell suspensions out of your tumor tissue. And this is also working perfectly for liver tumor samples. On the other hand, we are also offering a liver dissociation kit for mouse tissues, working also with pre-installed programs and the purple C tubes, and this was specifically engineered towards the isolation of liver sinusoidal endothelial cells, L-sacs, and Kupffer cells. Yield and purity is here absolutely similar to what you get out of conventional perfusion procedures. But this liver dissociation kit has one big limitation. When you work with this kit, you will not isolate functional hepatocytes or stellate cells because the shearing forces within the C-tube and the pre-installed program are just too high to result in viable hepatocytes and hepatic stellate cells population. This specific limitation, not being able to get viable primary hepatocytes out of liver tissue, was the basis to start the development of our GentleMax perfusion technology. And this technology I want to present to you in detail within the next slides. Isolating primary hepatocytes from liver tissue is a very complicated, complex, and challenging procedure. And this is what I want to show you on this slide, how this is conventionally done. If you do it conventionally, you normally work with narcoticized animal, and this means you always require an official animal experimentation approval by the different authorities. But let's have a little bit more precise look on this complex experimental setup. These are pictures taken from a publication from a lab from Stanford University, but I believe this is how it looks in a lot of different labs all over the world. Here on the right side, you have a water bath set at 42 degrees Celsius with all the perfusion liquids inside. Then next, you have your peristaltic pump with adjustable speed, which ensures that you generate a flow and pump the liquids into your animal. And here on the surgical table, you have your little needle you need for cannulation. How is this cannulation done? So basically you have, like I said, your narcoticized animal. You open up the belly of this animal, and then you have to cannulate the inferior vena cava with this little needle. And this is already very, very challenging. You can see here how small and tiny this is, and you really have to hit it perfectly. Afterwards, you have to adjust the speed of the peristaltic pump in a correct way, and then you normally perfuse with buffer to remove the blood. This you can see here in the picture B, how the organ is basically a little bit swollen because you pump a lot of liquid in it, and it's getting way more pale because you pump the blood out. And after the perfusion with the buffer, you perfuse or do the enzymatic digestion with raw collagenase. And after the liver is digested from the inside, the cell-cell contacts are digested, you will take out the liver and manually dissociate it so that you end up with a single cell suspension from this liver, mainly containing primary hepatocytes. So now we saw how this is conventionally done with this complex experimental setup, and I want to show you now how we do it with the GentleMax perfusion technology. First and most important, we work with resected organ from sacrificed animals. So no narcotization or manipulation of narcoticized animals. We do perfusion with only one liver lobe. If we talk about mouse liver perfusion, we take the biggest liver lobe, the left lateral. If we talk about rat liver perfusion, we normally take a smaller one, for example, the right dorsal liver lobe. You don't need to do any cannulation of blood vessels because we have an array of 4 needles which spike in to the organ at a random position. The complete program and procedure is automated with preset programs. It's very easy, convenient, and very fast to learn. And because we also use our own enzymes and reagents, it's very reproducible with lot-to-lot consistency. So what do you need in your lab to be able to perform the GentleMax perfusion technology. Of course, you need to have the instrument, the GentleMax octodissociator with heaters. If you already have one in your lab, probably you need to have a perfusion software update, but this is freely available online. You simply download it, put it onto a USB stick and update your program. Moreover, you have to buy the GentleMax perfusion sleeves depicted here at the bottom. These are our new adapters which are needed for the perfusion. Moreover, you have some running costs, which are the consumable and the reagents, and this is our new perfusion tube, the GentleMax perfusers, and our Max liver perfusion kit for mouse and rat, having the buffer you need, the reagents you need, and the enzymes you need. The centerpiece of this new technology is our new perfusion tube, the GentleMax Perfuser. This new tube has an inbuilt peristaltic pump. Let's have a little bit more precise look on it. It comes in 4 pieces, so you have the base with a needle array and the inbuilt peristaltic pump. You have a grid and a clamp. These are basically the 2 pieces you take at the beginning to place and secure your liver tissue, and then you have the top part, the lid, which has an adjuster with scale, which you can use to adapt the penetration depth of the needle, and you have 2 Leerlock connectors, which are used to aspirate used buffer or to add fresh one. And the peristaltic pump of the new perfuser tube is powered by our new adapter, the GentleMax perfusion sleeve, which is depicted on the slide here. What you can see on this image here on the right is a top view onto the instrument. This here on the left, it's the standard sleeve you would normally use for C and M tubes, and here above it you have our new perfusion sleeve. And this exchange of the sleeves really just takes some seconds, and then your instrument is ready to go to perform perfusion. So I was now already probably telling you 4 or 5 times the word peristaltic pump, but how is this actually working? How is the pumping action being performed inside the new perfusion tube? And why do we not need any kind of cannulation? So very important is that we have a liver lobe with an intact capsule. And we basically use it as a kind of balloon. So we take the array of the 4 needles, which is basically here in blue at the schematic drawing, and spike in to this liver lobe anywhere. And since these needles are inside the tube and we have a capsule around, we can use it as I said, as a kind of balloon and pump in different liquids by overpressure, and then they will nicely distribute inside the tissue and perform the perfusion. How is this peristaltic pump working? So you have one opening here to the base, so that the liquid which is in the base can get sucked in, and then you have a pumping channel which is constantly deformed by the action and rotation of the perfusion sleeve, thereby generating a flow. And basically, the liquids which are sucked in here are redirected via the pumping membrane into the needles and then into the tissue. And what is pumped into your tissue via this peristaltic pump? Basically, different buffers at different steps of the protocol. All of these are made from the buffer concentrate. You have washing buffers, you have equilibration buffers, but of course you also have buffers for the different enzymes. Moreover, we have different reagents in our out-of-the-shelf kits for best enzymatic activity and optional tissue storage. And very importantly, we use a combination of 3 different enzymes with lot-to-lot consistency, and this is a big difference to the conventional liver perfusion, where you would normally only use raw collagenase. And of course, these 3 enzymes with lot-to-lot consistency and also all these pre-formulated buffers and reagents make the perfusion very reproducible and reliable. Now you got to know all the different players we need for the GentleMax perfusion technology. But how does the workflow of perfusing rodent livers and isolating primary hepatocytes really work? I will take you step by step through this workflow in the next slides and show you how easy and convenient this new technology is. It all starts with a resected liver from a sacrificed animal and the assembly of the perfuser. And this is a very critical step because it's time sensitive. Ideally, it takes 5 to 10 minutes after sacrificing the animal to resect the liver and assemble the perfuser. Because the longer you wait, the blood will start coagulating, will form clumps, and will hinder perfusion dramatically, and yield and viability of your isolated hepatocytes will drop. I know it's really hard sometimes to keep this time frame, especially if, for example, your animal housing is not in the same building as your lab, or if you receive tissue samples from a cooperation partner. We also have a solution for that. You can use our Max Tissue Storage solution plus reagent E, which is part of the liver perfusion kit, and then you are fine up to 2 hours without loss of viability or yield. Let's have a more precise look on how to assemble this perfuser in detail. So you have your resected liver lobe, still red and full of blood, place it in the middle of the grid, then take the clamp and attach it from above to really fix the liver lobe in its position. Next, you will take the lid from the already installed perfuser on the instrument, click it into the grid clamp assembly, then transfer it back to the perfuser base. Just drop it, turn it slowly counterclockwise till it drops to a lower position, and then close it by turning clockwise. And this is already the perfect position because by closing the lid, the 4 needles from the needle array will already penetrate the tissue. And for mouse, you don't have to touch the adjuster and adjust the penetration of the needles. It will have the perfect position and will just spike into the organ. The perfusion process itself on the device in the perfuser tube takes around 25 minutes. It's a preset program. It consists out of different steps of the program. So you have a priming in the very beginning, 5 seconds to get all the air out of the system. Then you would install the lid with the attached grid clamp and the liver piece inside. You will have 4 short-term washing cycles of 30 seconds, 1 long washing cycle with 13 minutes, a short round of equilibration with the enzyme buffer, and then our enzymatic digestion, 10 minutes for mouse liver, 12.5 minutes for rat liver. During the different steps, you have to aspirate the used buffer and add new buffer. Here also a short video sequence to show you how it's done. So basically you can go in with a glass Pasteur pipette, connect it to a suction pump and simply suck out the used buffer, and then via the lure lock connectors add new buffer. The cool feature here also, theoretically you could save all the used buffer and all the flow through of the liver which was generated during the perfusion to use for downstream analysis. After you have performed the perfusion process in the perfuser tube, you remove the perfused liver lobe from the perfuser, add it to a C-tube and do the hepatocyte release in a C-tube in a different program. Let's also have a look into a short video sequence to see how it's done. So after the program, you will remove everything. What you can directly see when you pull out the tube is that the liver is also now nice and pale. You will save the used enzymes for the round of the hepatocyte release in the C-tube. Just lift the clamp, take out the perfused liver. Typical appearance of a perfused liver, nice, pale, all the blood is out. You put it into a C-tube, close the C-tube, attach it upside down on the device on a position equipped with a regular sleeve, and then start your hepatocyte release program. Here you can still see the intact organ, But what you can see is that already after 5 to 10 seconds, the organ is dissolved into single cells. So you just apply minimal mechanical shearing here and directly dissolves into single cell suspension with highly viable hepatocytes. Last step would be then the purification of your hepatocytes. Normally you would do this with low spin centrifugation steps. Optional, you could also do a debris removal step. For this, we have a special 10-minute density gradient centrifugation program to really get ultra pure hepatocytes with our debris removal solution. The cool thing here is you can use full acceleration and full brake, which will really save you time, and you end up with ultra pure hepatocytes. For example, if you need them for downstream sequencing or genomic analysis. Taken all together from sacrificing the animal until you have your purified hepatocytes and also the NPCs in the supernatant, it takes 45 minutes. And this is basically for one sample. But since you have 8 positions, in theory, you could do 8 perfusions at the same time, which means you have 8 perfusions done within 45 minutes. And this is how a typical suspension looks after low spin centrifugation, after perfusing with the GentleMax perfusion technology. So for example, this is the result from a perfusion of a left lateral liver lobe of a healthy female black 6 mouse, 8 weeks old. So you have pelleted hepatocytes. Normally you end up around 10 million cells per left lateral liver lobe. And in the supernatants, you have all the different non-parenchymal cells, LSACs, Kupfer cells, but also stellate cells, and cholangiocytes, around 5 million cells. So let's talk about yield and viability. Like I said, from a mouse left lateral lobe, you can expect 10 million hepatocytes with above 85% viability. If we talk rat, I told you here we use a smaller liver lobe, for example, the right dorsal lobe, where we get more than 20 million hepatocytes, also with 85% viability. Viability here was measured with flow cytometer. Now we have our primary hepatocytes. Let's see how they behave in culture and if we can compare them to conventionally isolated primary hepatocytes. And this comparison of conventionally isolated hepatocytes versus GentleMax perfusion technology isolated ones were already done by some cooperation partners together with our R&D. This was published last year in a peer-reviewed magazine. Our cooperation partners here were Ralf Weiskirchen from the Uniklinik Aachen and Martin Roderfeld from the Universität Gießen. Let's have first a look of what they got out of the perfusion with the GentleMax perfusion technology in terms of yield. So here we have 31 different mice, which were mainly bulb C and black 6 mice, 6 to 12 weeks old, and hepatocyte yield was pretty stable above the 10 million hepatocytes per left lateral liver lobe. Here they even end up at 34 million per gram of liver tissue, which would equal into more than 20 million per left lateral liver lobe. And also viability here, also above 85%, close to 90% hepatocyte viability in average. The appearance of the liver lobes before and after perfusion show a typical color change. Here on the left, you see the very, very reddish perfusion, before perfusion liver lobes filled still with blood, and then after perfusion, first the washing with the different buffers, you have all the blood removed, and then you do your enzymatic digestion. You have the typical change from reddish to a pale color, which shows you that the perfusion is really working and the tissues are nicely perfused. Afterwards, if you then isolate the primary hepatocytes out of these perfused organs, you can nicely culture them in cell culture. Here are pictures after 24 hours, for example. They show their characteristic flattened hexagonal appearance and also multinucleated, another typical sign of hepatocytes in culture. After plating and culturing these hepatocytes, the researchers in this publication also analyzed the characteristics of these cells, here in this case by immunofluorescence staining. They stain for cytokeratin 8 and the tight junctions marker Z01. What you can see is that the cells are nicely polarized, also show the typical binucleated, multinucleated appearance of hepatocytes, but more important, also you will find a strong expression of hepatic nuclear factor 4 alpha, which is the master regulator of hepatic differentiation, really showing you that these are hepatocytes growing in culture here. And the authors also did some biochemical feature analysis of the different isolated hepatocytes. What you can see here on the left is a qualitative mRNA expression analysis. You have 3 different sets of samples. You have normal liver tissue, you have hepatocytes isolated with the GentleMax perfusion technology, and conventionally isolated hepatocytes. And what you can see here is that all of them express one of the main markers of hepatocytes, albumin. And again, you will find a positive signal of hepatic nuclear factor 4 alpha in all of the different sets, showing you that there is basically no differences between liver tissue, GentleMax, isolated and conventionally isolated hepatocytes. Moreover, they did some stimulation experiments with the pro-inflammatory cytokine interleukin-1 beta, which is known to induce lipocalin-2 expression. And what you can see here is without the interleukin, lipocalin-2 expression is very low, and you clearly see an induction of the lipocalin-2 expression after the addition of interleukin-1 beta, which clearly shows you that both of the hepatocytes, conventionally or GentleMax technology isolated one, are sensitive to interleukin-1 beta and can be stimulated. So this was a quick ride through the GentleMax perfusion technology. Let's sum up what we have learned and have an outlook. In summary, we were not able to find any differences in appearance and behavior between hepatocytes isolated with the GentleMax perfusion technology and the conventional method. Functionally, both of the differently isolated hepatocytes were identical. But you will have several advantages when working with the GentleMax perfusion technology. In my eyes, the biggest advantage is that we work here with resected rodent liver. This means you don't need any kind of narcotization or manipulation of narcoticized animals anymore. If you do the conventional perfusion, you will do it in situ. All the other organs will also get perfused. Here, we take the resected liver, take one liver lobe, and do ex vivo perfusion, which means all the other organs of the sacrificed animal can be used for other purposes. Moreover, we don't need any kind of cannulation, and this gives us also way more flexibility towards the age of the mouse, because especially young mice are very, very hard to cannulate. Moreover, we only perfuse one liver lobe. If we talk about mice, we perfuse the biggest one, the left lateral liver lobe, and this leaves the rest of the liver, which is approximately 60%, for other purposes. So for example, you can take one resected liver, take one liver lobe for the perfusion and get your primary hepatocytes, and can take the rest of the liver for validation studies or, for example, doing spatial biology. And all these points also promote the 3R principle: replacement, reduction, refinement of animal experimentation. If you have worked with frozen hepatocytes before, here you have now the chance to work with fresh hepatocytes isolated from your own mouse models. Additionally, the GentleMax perfusion technology is automated, and this makes this technology reproducible, standardized, and operator independent. Moreover, it's very easy to use, so it's very fast to learn. You don't need any kind of extensive training for several years. I even have some customers where the bachelor students now start isolating their own primary hepatocytes from liver tissue with the GentleMax technology, and this was something which was not possible before. Moreover, when you use our Max Liver perfusion kit, the reagents which are included will have lot-to-lot consistency, and that fosters standardization and reproducibility again by using the combination of 3 enzymes instead of raw collagenase. Also, the digestion will be more gentle and efficient. Let's have a look at the instrument and the consumables. They work in a closed and sterile system, which reduces the risk of microbial contaminations. Moreover, you are able to collect the perfusion liquids when exchanging the buffers during the different washing steps or before the enzyme digestion, which would be not possible when doing it the conventional way. If you use the GentleMax Octo dissociator with heaters, you have 8 positions and can perform 8 perfusions in parallel at the same time. And this really boosts the output of your perfusion experiments. Last but not least, if you use the C-tube for the tissue dissociation after the perfusion, from the perfused liver tissue will make your output way more reproducible than doing it manually by mincing the perfused tissue. I hope you enjoyed the last 30 minutes and found out everything you wanted to know about the GentleMax perfusion technologies. We are more than happy to show it live in your labs. Demo requests can be directly directed to my dear colleague Elahe, so it's elahes@miltenyi.com, or you simply go to the miltenyibiotec.com website homepage and look into the Max sample preparation portfolio. There we also have one lead form where you can directly request a demo of the GentleMax perfusion technology. Moreover, we have a lot of how-to videos on YouTube. Also, most of the video sequences I showed you earlier in this webinar can be found on YouTube, especially now for the liver perfusion. We have, I believe, 9-minute long how-to video showing every single detail how to do it. Just Google liver perfusion Miltenyi and you will get this video. This brings me to my last slide. I want to thank you for your attention, but I also have to thank a lot of people at Miltenyi for helping developing this fascinating new perfusion technology, especially our R&D department, our plastics department, automation, software, production, and of course, all the beta testers and early adapters who helped us really to develop this technology further. And again, thank you for your attention, and I'm happy to take your questions.